Hello, everyone. Welcome back to my video tutorial for single-cell T-cell receptor sequencing dataset analysis. In my previous video tutorial, I showed you how to process the 3K demonstration dataset from the Scirpy package. In today's video tutorial, we are going to define the clonotypes and the clonotype clusters. So first, let's define the clonotypes. To define the clonotypes, first we need to calculate the nucleotide sequence-based distance matrix for all the VJ sequence and VDJ sequence in this dataset. You can see here we didn't set any argument in the distance calculation function. So by default, we will calculate the nucleotide sequence-based distance matrix. So we can calculate the default nucleotide sequence-based distance matrix now. Then we can define the clonotypes by nucleotide sequence identity. Let's define the clonotypes. So in order to visualize the clonotypes, Scirpy uses a network-based approach for clonotype definition. So we can compute the layout of the clonotype network now. After that, we can plot the clonotypes by different parameters. On this demonstration, we can plot the clonotype network using the cell source. In the metadata, we have different cell source type, patient's information, and sample information. You can see we plotted the clonotype network colored by the cell source. You can see we have cells from blood samples. They are in blue color. Cells from normal adjacent tissue, they are in pink color, and cells from tumor are in green color. So in this figure, you can see lots of dots on the figure. The dot sides represent the cell numbers that have the same clonotype. And the numbers next to each dot indicates the clonotype ID that we saved in the TCR sequencing data. And also in each dot you can see it indicated by the pie chart to show the clonotype's origin from different tissue. So here we defined the clonotypes based on the nucleotide sequence. We know even the nucleotide sequence is different, but a different nucleotide sequence set can be translated into the same amino acid sequence. So we can define the clonotypes clusters based on the amino acid sequence similarity. So first we need to recompute the clonotype network based on the amino acid sequence similarity. So let's calculate the distance based on the amino acid sequence distance. Let's run. Okay, after the distance calculation, we can define the clonotype clusters based on the similarity of the scTCR-seq sequence. We can use the define clonotype cluster function to define the clonotype clusters. Let's run. So we defined the clonotype clusters based on the similarity of their scTCR-seq sequence. Again, we need to calculate the clonotype networks. Then we can visualize the clonotypes. You can see here, now we set the argument as sequence equals amino acid. In previous calculation for the nucleotide sequence, we use the default settings. So now we can compute the layout of Clonotype network. After that, we can plot the Clonotype clusters. If we run, you can see we generated a figure to show the Clonotype clusters. Here we visualize the Clonotypes according to the patients. You can see now there are connections between different Clonotypes. Because we set the cutoff as 15 when we calculate the distance, so any Clonotype has a distance below 15, we will see a connection between different Clonotypes. For example, here the cluster 159, it has 5 different Clonotypes, but they have been defined as the same Clonotype clusters because they have the same amino acid sequence in one of the VDJ sequence. For example, we can extract the amino acid sequence information from different Clonotype clusters. We can use the cluster 159 as an example to extract the information. You can see here, we are going to use cluster 159 as an example to extract the VJ1 junction amino acid information, VDJ1 junction, VJ2 junction, VDJ2 junction, and the receptor subtype information from this cell cluster. So let's subset the data for the cluster 159. Then we can have a look at the information we extracted from the cluster 159. Let's run. You can see now we have the information for the cluster 159. You can see we have 5 Clonotypes here. They have been defined as the same Clonotype cluster 159 because they have the same amino acid in the VDJ1 junction. You can see for the VJ1 junction amino acid, the first 3 are the same, but they are different from Clonotype 4 and 5. You can see here for the VJ2 junction amino acid, they are also different, but they have the same receptor subtype because in this dataset, we only have TRA and TRB receptor subtypes. Here are the cell numbers for each Clonotype. So you can see in the cluster 159, we have 26 cells have the same Clonotype cluster. So you can see here we cluster the Clonotypes as the same amino acid sequence, but even they are in the same cluster, they have different VJ1 sequence. So you can also include the V genes in the Clonotype definition. For example, we can run the define Clonotype cluster function with the same V gene argument as true. Then you can redefine the Clonotype clusters. Now the cluster should have the same V genes. Let's define the Clonotype clusters with the same V gene argument as true. So after defined the Clonotype clusters with the same V gene as true, now we can find out the Clonotypes in our previous analysis that have different V genes. Let's run this function to find out the clone clusters. You can see the Clonotype cluster 280 and 765 have different regions. When we defined the Clonotype clusters use the same region argument as true, then we will split both clusters into different subclusters. We can use this code to have a look at the subclusters from cluster 280 and cluster 765. You can see, following the analysis, use the same region as true. Now we split the cluster 280 into subcluster 280 and 788. The cluster 765 we split into cluster 765 and 1071. Okay, that's it for today's video tutorial. I showed you how to define the Clonotypes and also how to define the Clonotype clusters. In my next video tutorial, we are going to perform Clonotype analysis. I hope to see you in my next video tutorial. Thank you.